So welcome to Jumbo IS and welcome to lecture number 2 of Science and Technology in this lecture series called Prelims Powerplay 2026. Now today's lecture is going to be based on red biotechnology. Well, if you watch Science and Technology lecture number 1 properly, I hope you gave your 100% attention. So if you gave your 100% attention, then you must know this, that red biotechnology deals with biotechnology applications in the field of medicine. So whatever developments happened in India in the year of 2025 with respect to biotechnological applications in medicine, that is red biotechnology, will be covered in today's lecture. So all your current affairs with respect to red biotechnology, all medicine related biotechnological applications, news items, current affairs that were related to, well, biotechnology in medicine in this year 2025 will be covered in this lecture. Okay, so red biotechnology is the topic. So let's straight away dive into today's lecture. Okay, without any further introduction, I won't bore you with any more introduction. Let's straight away dive into red biotechnology. Okay, so Science and Technology lecture number 2, red biotechnology, and this is that biotechnology that deals with the field of medicine. Okay, now in today's lecture we are going to basically cover 6 topics. Okay, what are those 6 topics? Let's take a look at that. So number 1 topic is, well, CAR T cell therapy. It is a therapy which uses biotechnology to cure cancer, particular forms of cancer. Okay, and India also developed some indigenous technology using CAR T cell therapy. So we'll talk about CAR T cell therapy. Then topic number 2 is related to gene therapy. So we'll talk about gene therapy. Okay, and various applications in India. Then topic number 3 is called personalized medicine. What is personalized medicine? Well, each medicine tailored to your own DNA, because we all have different DNA and we all respond to different medicines differently. Some medicines work on some people, some medicines don't work on some people, and some people are allergic to certain medicines, right? It's all because of your genotype and phenotypes. Okay, so we'll talk about personalized medicine. Then topic number 4, because in this year many cancer curing vaccines were developed. So this would be next topic. 5th topic would be related to biopharmaceuticals. Pharmaceutical interventions, medicines, vaccines, all these things developed via biotechnology. And in the end, we'll talk about futuristic biotechnology, which is use of AI and 3D printing in biotechnology. Okay, how can we cure people using AI and 3D printing? So these are the topics that we shall zero in our attention on for today's lecture. So let's start discussing them one by one. Okay, starting with topic number 1, and that is CAR T cell therapy. Okay, so CAR T cell therapy is basically a therapy that cures cancer. Now you all know what cancer is, right? Cancer is a situation in which cells of your body multiply. Okay, they multiply exceedingly. Okay, I mean, they multiply a lot, more than required. And that is why tumors are formed. And when those tumors become extremely large, well, then, well, certain death is also possible. It is basically very dangerous, right? Now, these cells which multiply a lot are known as cancerous cells. And if they happen in your stomach, if these cells occur in your stomach, then it's stomach cancer, liver cancer, blood cancer, right? So it depends where these cells are and which type of cells, have become cancerous. Okay. Now, when someone gets cancer, what are the cures available? This is very important, and this is where CAR T cell therapy comes in. So whenever someone is diagnosed with cancer, well, generally people are advised chemotherapy. So in chemotherapy, you inject a lot of medicines into a person. Those medicines kill cancerous cells. Then there is another therapy called radiotherapy, in which you expose a person's body with radiations. Those radiations will kill cancerous cells. But the problem with both chemotherapy and radiotherapy is that these interventions do kill cancerous cells, but they also kill normal cells also. Radiations are powerful. They also affect normal cells. Medicines in chemotherapy are powerful. They also affect normal cells. And that is why there are a lot of side effects of chemotherapy and radiotherapy. You must have heard about it, right? A person undergoing chemotherapy, well, becomes bald. There are a lot of side effects. It's a very painful therapy. We all have heard about it, right? That's because both these therapies also affect normal cells. What if there is a therapy that only specifically targets cancer cells, doesn't affect normal cells at all, only specifically targets cancer cells? And what if that therapy doesn't require any medicines in your body, doesn't require any radiations in your body. What if you can genetically engineer your own immune system? You have an immune system, that immune system attacks foreign bacteria, viruses, right? What if you genetically engineer your own immune system to attack only cancerous cells? No foreign substance, no foreign radiation is added. Your own immune system is genetically engineered to kill only cancer cells, right? That would be great. Yes, this is what CAR T cell therapy is. It genetically engineers, trains your immune system to attack cancer cells and cancer cells alone. Okay, so let's talk about this, and then we'll talk about what sort of new medicine has been created in India, by India, using CAR T cell therapy. Okay, so let's begin. So CAR T cell therapy. So about the therapy, well, you have your immune system. In your immune system, there are these specific cells, immune cells, immunity cells, which are known as T cells. Okay, so T cells are those cells which are responsible for your immune system. Okay, also known as T lymphocytes. Clear? Now, in CAR T cell therapy, well, it is a form of personalized immunotherapy. So there was chemotherapy, there was radiotherapy. Immunotherapy is when you use your own immune system to kill cancer. That is immunotherapy. So it is a form of personalized immunotherapy, and I'll tell you how it is personalized, in which a patient's own T cells are genetically engineered to recognize and kill cancer cells. Okay, so why is it personalized? Because we take your own immune cell system, your own immunity cells. We don't introduce foreign immunity cells in your body. No, your own immune system is programmed. So it's highly personalized also. Okay, the full form of CAR T cell is, well, we all understand what T cell is. CAR stands for chimeric antigen receptor. Okay, chimeric antigen receptor. Now, to understand and to be able to remember this full form, well, just understand the meaning of this word antigen. So the thing is, I told you that these T cells are programmed to identify only cancer cells and kill them. Okay, how do they identify these cancer cells? Every cancer cell has a specific protein on it, okay, on its surface. That specific protein is called antigen. Okay, so when you program these T cells, okay, now they will identify this antigen on cancer cells only, because these antigens are only found on cancer cells. They will attach on it and destroy it. Okay, so you program these T cells to identify this specific antigen. This is CAR T cell therapy. It was discovered in 2017 and it has been path-breaking. Okay, now T cells are modified to express an artificial receptor that binds a specific antigen on tumor cells. So all these T cells have a receptor, and this is an antigen on cancer cells. It specifically fits onto this antigen lock. Okay, and that's how it destroys that particular cancer cell, right? And that antigen is only found on cancer cells, not normal cells. So there is, well, very little side effects, very less number of side effects, because normal cells are, well, left out scot-free. They are saved. Okay. Now, based on CAR T cell therapy, India developed its own therapy called NexCAR19. Is India's first indigenously developed commercially approved CAR T cell product for certain relapsed refractory B-cell leukemia, a form of cancer, and lymphomas. So these are the two forms of blood cancers, lymphomas and leukemias. NexCAR19, which is based on CAR T cell therapy, targets these cancers. Now, what is the meaning of these two words, relapsed or refractory? We'll talk about it, don't worry. Okay. Now, what is the process, the entire process of CAR T cell therapy? Well, the first step is leukapheresis. Remember this word, leukapheresis. What happens in this step? Well, T cells are collected from patient's blood. So you extract T cells from a patient's blood. I'm so sorry. Okay, so T cells are extracted. Then step 2, genetic modification. In a lab, you modify these T cells. Okay, then step number 3, you increase the number of these T cells. These genetically modified cells are amplified, expanded. Their numbers are increased. This is expansion. Then, well, you remove normal T cells from a person's body so that these new genetically programmed T cells can be injected. Okay, so this is known as lymphodepleting chemotherapy. Before infusion, before injecting these genetically modified T cells, patients usually receive chemotherapy to kill Normal T cells. So normal T cells are killed. Now new T cells are added. This is CAR T infusion. Do you know what? Why is this therapy better? Because in chemotherapy, once you're given chemotherapy, once the medicine is flushed out of your system, well, then nothing can happen. What if your cancer reoccurs? You have no medicine in your body to kill that cancer. But in CAR T cell therapy, well, your immune system has now been changed. So even if in future the cancer reoccurs, well, your immune system is prepared to kill it again, again and again. That is why this therapy is known as a living drug. It lives in your system forever. So these living drug cells can persist for months to years, providing ongoing immunosurveillance, ongoing therapy, okay, and reduces relapse risk. Relapse means when cancer reoccurs. Okay, remember this term. CAR T cell therapy is called living drug therapy. Okay, the drug lives forever. Okay, not forever, but for a long time, because these are cells, your own cells. You understand? Now, what are the benefits of this therapy? Well, number one, better response and remission rates. Okay, when cancer starts to recede, this is known as cancer remission. So, good response. Number two, living drug, long-term protection. Number three, chemotherapy and radiotherapy have side effects because they harm normal cells also. This doesn't. And number four, it is personalized medicine. How is it personalized medicine? Well, it is personalized because we are taking the patient's T cells, modifying them and introducing it into his body. Okay, so it is targeted. Targeted why? Because it only kills cancer cells. Targeted personalized medicine. You understand the point now? Okay. But the thing is that this therapy also has certain challenges. Okay. Number one challenge is there are some side effects. Okay. Number one challenge is cytokine release syndrome. You see, the thing is that when you modify a particular T cell, sometimes there is an adverse reaction. Every immunity cell, T cell, releases a protein called cytokine. This cytokine basically decides your immunity response. Sometimes these T cells, which have become genetically engineered, they become hyperactive. They release a lot of cytokine. When they release a lot of cytokine, your immune system becomes hyperactive, causes a lot of inflammation, and even organ inflammation can cause can destroy other organs as well. This is known as a cytokine storm. It is rare, but it is possible, a possible side effect. Okay. Then sometimes immune effector cell associated neurotoxicity. Sometimes your immune system destroys your nervous system. These modified T cells can impact your nervous system, can cause neurological symptoms, confusion, encephalopathy. Encephalopathy means when your brain is impaired. Okay. And then other side effects or other challenges in this therapy are, well, if by just a stroke of luck or by 1% chance, if a particular antigen on a cancerous cell escapes the detection, then the cancer can relapse. So this is there. Okay. It is only effective in blood cancers, not effective against solid tumors. There are certain reasons for it. Plus, it is very difficult to make this therapy, manufacturing complexity, and very high cost. 400 to 500,000 US dollars, which is around, let's suppose, 7-8 crores. Okay. Now, not 7-8, 4-5 crores. So this is a problem. Okay. So very high cost. By the way, key terms like cytokine and encephalopathy are given in your notes, the definition, but I've explained it to you, so I hope you understand. Okay. And then about Indian therapy, well, India developed NextCAR therapy based on CAR T cell therapy. Okay, it was developed by ImmunoAct startup, incubated by IIT Bombay and Tata Memorial Center. Okay, it is approved by Drug Controller General of India, Central Drug Standards Control Organization. It treats lymphomas and lymphoblastic leukemia. Happens to children. Okay. And yes, remember I told you CAR T cell therapy is better for relapsed or refractory cancer. So relapsed cancer is which reoccurs. After treatment, you become better and then it reoccurs. Refractory is when your cancer treatment is not responding. You are given chemotherapy, but nothing is happening. Cancer is still growing. That is refractory cancer. When your body doesn't respond to treatment. Okay. Now, NextCAR has certain benefits. Okay. CAR T cell therapy is expensive, but India's NextCAR is cheap. So 1/10 of the cost of actual CAR T cell therapy. Plus it has shown various benefits like treatment benefits, people have recovered. Okay, plus it has a very big cost advantage. Next CAR is cheaper than Western CAR T cell therapy. Okay, you understand this point? So that's about it. All right. So let me just stop this lecture for a few seconds because I want to share something really important. Look, along with this lecture series, you will also get notes, exhaustive notes, which you have to memorize. Then you will be enrolled in our WhatsApp groups where you can ask all your doubts, even the simplest of queries. And along with this lecture series, you will also be getting a test series, an exhaustive list of tests tailor-made for your UPSC Prelims 2026 examination. To register, to get access to notes, WhatsApp groups, and tests, you would have to register on our app or on our website. The links to both app and website are given in the comment section below. You just have to click on the link and it will take you to our registration page. So register for Prelims Powerplay 2026 because you have no idea how beneficial this lecture series is and you'll just be spending 1 hour of your day. So join now, click on the links given in the comment section below. Now let's quickly shift our focus to next news item, and that is gene therapy. Now listen to me very carefully, okay? You see, certain diseases are genetic, that is by birth. That is, they didn't occur to you because of any infection, bacterial or virus, or because of any lifestyle disease. Like you were living a sedentary lifestyle, you were not exercising, so you got blood pressure, cholesterol, okay? These are lifestyle diseases. Some are infectious diseases. But some diseases are by birth. They are genetic diseases. It's not your fault. Your DNA was programmed in such a way that you will have this disease. There are many genetic diseases. Okay. Now, how can you cure them? Up till now, there was no cure for genetic diseases. Look, for bacteria and viruses, you have antibiotics and antivirals. For lifestyle diseases, you improve your lifestyle, and then there are certain other treatments. For genetic abnormalities, well, what was the treatment? The treatment was only symptomatic. You only reduce the symptoms, but you do not cure the root cause of the disease. Only symptomatic treatment was available. But now, with advancements in genetic engineering, now you can address the root cause also. You change that person's DNA. You first identify which portion of your DNA is causing this disease. That identification is also very difficult. But now we have technology to identify that particular DNA segment, which will tell you that it is because of this that this disease has occurred, right? You identify it, remove it, replace it with normal gene copies, or just remove it. Okay? Once you remove it, well, the DNA that was causing that disease has now been removed. The person is cured. Now, this is called gene therapy. It's therapy because people are getting cured from a disease. And this gene therapy occurs through genetic editing. You edit a person's DNA, right? So there have been some developments with respect to gene therapy in India as well as the world, okay, in this past 1 year. So let's cover all of them, okay? So gene therapy. So about gene therapy. So gene therapy modifies a patient's genes to treat or prevent diseases by addressing the underlying genetic defects rather than symptoms, okay? Now, gene therapy targets the root cause, okay, such as a single gene conditions. Only one gene in your DNA was problematic. You replace it. That is called monogenic. Sometimes 2-3 genes are responsible for that abnormality. That is polygenic. Nonetheless, some examples are sickle cell anemia, hemophilia. These are all blood diseases. Okay. Now, what are the techniques used in gene therapy? So techniques include gene addition. So sometimes you have a disease because you don't have one particular gene in your DNA. Sometimes maybe I'm having a genetic disease because other people have that gene, but I don't have that gene. Because of lack of that gene, I'm having this disease. So you add that gene. Okay, this is known as gene addition or gene silencing. Sometimes I have an extra gene and others don't, and that is why I'm having this disease. So that gene can be silenced, inactivated. This is gene silencing. And sometimes it's very simple. You cut the faulty gene, remove it, and replace it with normal copies of genes. Okay, this is gene editing, proper gene editing. And for that, we use now there is a very good technique for gene editing. This is known as CRISPR-Cas9. Most of you already know about this. Remember, CRISPR-Cas9 technique is a revolutionary technique for gene editing, which has made gene editing simple and cheap also. So now all gene editing is done through CRISPR. Okay, it's very simple. Delivery often, yes. So in case of gene addition or gene editing also, You have to introduce normal copies of genes. So how do you introduce normal copies of genes? Well, you use viruses, inactivated viruses that won't cause any infection, but would carry that normal gene into your body. They act as carriers. So these carriers are known as vectors. Okay, simple. Now, types of gene therapy. One is somatic cell gene therapy. In this, your non-reproductive cells, which are called somatic cells, remember this term. Somatic cells are non-reproductive cells. They are edited. Their DNA is edited. Okay. So when you edit somatic cells, what happens is that your offspring will not have that edited function. It won't get transferred to your offspring because you are not editing reproductive cells. It is permissible. Then there is germline gene therapy. You edit reproductive cells like eggs in case of females, sperms in case of males, embryos. When you edit germline cells, well, then that edited version will also be transmitted to your offspring. This is not permissible in many countries because many religious groups say that you are interfering in the work of God. So this is there. Okay. Now, how do you do gene editing? There are a few approaches. One is called, remember these terms, ex vivo. That's why I've introduced this. Ex vivo and in vivo. Now, ex vivo, cells removed, edited in a lab, and then reinfused. So your CAR T cell therapy and Casgevy, a UK therapy, UK-based therapy for cancer. Okay, CAR T cell therapy, Casgevy, these are ex vivo. You remove T cells, edit them in a lab, and then reintroduce. And there is an in vivo. You don't remove, you just introduce normal genes into the body. Genetic material delivered directly into the body. Okay, so ex vivo, in vivo. Just remember this. Now, delivery mechanisms. Mostly you deliver DNA through viral vectors, viruses. Okay, AAV viruses, these are called adeno associated viruses. Okay, no need to understand what are they. Just remember the name. AAVs, adeno associated viruses, and lentiviruses. These are the two types of viruses which are the best possible vectors for delivering DNA. Okay, and then there are non-viral vectors. Fat globules, sometimes you use, nanoparticles are used to deliver DNA, but this is very difficult and path-breaking research is required in this. This is easier and better understood by scientists also. So we use mostly viral vectors. Okay. Now some challenges in gene therapy. Well, high cost, immune reaction. Sometimes your immune system can react to this foreign DNA. So this is a problem. Or foreign vectors, viruses coming. Your immune system is trained to attack viruses. So this can happen. Sometimes off-target edits. Sometimes you wanted to target this cell, but you targeted the other. That can happen, risking cancer and short-term efficacy. So these are some of the challenges. By the way, you remember in Science and Technology lecture number 1, we talked about BioE3 policy. So in BioE3 policy, this gene therapy is one vertical. Okay, so yeah, that's about it. All right, and now let's quickly shift our focus to third news item, and that is personalized medicine. Now let's quickly cover this because most of us already know about this. So what is personalized medicine? Very simply put, every individual is different because we have a different genetic makeup. Okay, now because every individual is different, sometimes some people respond to a particular medicine, antibiotic, some people don't. Some people are allergic to a particular antibiotic, some people aren't. This information is very important for a doctor while prescribing a medicine. Yes, that is why. Now, generally, this information was not available. That is why we are moving towards personalized medicine. In personalized medicine, your medical therapy is tailor-made as per your individual makeup, genetic makeup. Okay? As per your DNA, we will prescribe the medical therapy to you. So it is personalized medicine. Now it is very difficult. Why? First of all, you need to know the genetic makeup of each and every individual. That is very difficult because our DNAs are very complicated. Each and every gene in our DNA has a specific function and it's very difficult to study them all. Clear? So that is there. Plus, we need to know, first of all, you have to identify all the genes. Then you need to know what each gene does. What is the function of each and every gene? And then you need to understand whether this medicine is applicable on this gene or not. Right? All these things are really complicated, but science is advancing in that direction where all of this is possible. And India has made certain developments in this. So let's talk about this. Okay. So personalized medicine. So personalized medicine tailors treatments to individuals' genetic, molecular, and environmental profile. Okay, the person's environmental circumstances are also there. Now, a branch of personalized medicine is precision biotherapeutics, targeted therapy, like CAR T cell therapy. It was targeted. So this comes under precision biotherapeutics. Represent advanced gene-based therapy like CRISPR, CAR T, mRNA vaccines. Okay, remember mRNA vaccines during COVID. All of this is precision biotherapeutics. Now, personalized medicine uses pharmacogenomics. Remember this term, pharmacogenomics. Pharmacogenomics is the science of studying how different medicines interact with different DNA. Because if you'll know this, then you'll know for sure whether this person will respond to this medicine or this person is allergic to this medicine or not. Okay, this is pharmacogenomics. You identify certain biomarkers in a person. Okay, so for example, there is a specific protein in your body which will not respond to this antibiotic. So these are biomarkers, and you use AI in all of this. Okay, because it's very complicated to predict drug response and customize care. We will shift from one size fits all. Whenever someone has cough, you give them azithromycin. That's one size fits all. Okay, we'll shift from that to personalized medicine. Okay, precision biotherapeutics, a BioE3 policy vertical, include genome profiling. Genome profiling means you need to do genome mapping, genome sequencing, understanding the DNA, entire genome of a person. For that, India has launched various projects also. Okay, and details about those projects like Genome India project, Indigen project are given in your notes. Clear? India's genetic diversity has been captured because Indians' DNA is a little bit different to European DNA. Then why are we following European medicines? Right? You understand the point. That is why India conducted its own genomic survey, which is Genome India project or Indigen project, to understand Indians' DNA better and then develop medicines on it. Okay. Now we support 100% FDI in this. PLI schemes are there. Under BioE3 policy, we have set up many biofoundries also. Remember biofoundries from previous lecture. So this is there. Clear? Then various technologies and their examples. Okay, genome profiling for that Indigen project, gene editing. Okay, this table is given in your notes, so go through this as well. Clear? There are some challenges with respect to this personalized medicine. First of all, very high cost, and we do not have the required infrastructure in India. But BioE3 policy is focusing on that. So yeah, go through this as well. All right. And now let's quickly shift our focus to next topic, and that is cancer vaccines. Using gene therapy and genetic editing and many other genetic engineering, forms of genetic engineering, we have developed cancer vaccines also. Okay. Now, one particular form of cancer vaccine is mRNA vaccine. mRNA vaccine was used in COVID also. How did mRNA vaccine work in COVID? Well, the thing was that what do vaccines do? Vaccines train your immune system to attack the virus. Yes. So COVID virus had one particular protein on it that was called spike protein. So what these mRNA vaccines did was, these mRNA vaccines injected RNA into your blood. That RNA instructed your body to produce spike proteins. Now, as your body started to produce spike proteins, that spike protein was identified by your immune system and your immune system started attacking it. Okay? Now your immune system was trained to identify spike protein. Now, when actual COVID virus would come, it will also have spike protein. Your immune system will attack it. So now you're immune. Right? Similarly, mRNA vaccines can be created to attack cancer cells also. Right? This has been done in UK. Clear? So let's talk about these cancer vaccines. Okay. So first, we'll talk about mRNA vaccine. You understand how mRNA vaccines work now? So these are cancer vaccines use synthetic messenger RNA. RNA is a protein in your body that instructs your body to produce other proteins. So this lab-produced RNA was introduced in your body to instruct your body to produce spike protein, that COVID protein, to produce tumor-specific antigens, proteins, training immune system to attack cancer cells. So these mRNA vaccines are trained to attack cancer. So global examples are Moderna and Russia's Enteromics. India leverages mRNA expertise from Gemcovac for oncology trials under BioE3 policy. India is also developing an anti-cancer vaccine under BioE3 policy. Okay. Challenge is again high cost, cold chain, because you need to store vaccines in cold chains, and scalability, mass production of these vaccines. Then there is another cancer vaccine which is not mRNA vaccine, but it cures one particular form of cancer, and that is called cervical cancer, which occurs only in girls. Cervical cancer is caused by a particular virus called human papilloma virus, HPV. So if your body is made immune from HPV virus, then this virus won't infect your body, you won't have cervical cancer. HPV vaccine was created by India recently. It was called Cervavac, produced by Serum Institute. Okay, and the Cervavac is now being given to young girls so that they're immune from what? Cervical cancer. Okay, so this is the thing. Go through this very quickly. All right, and that would be all. But there are two more things. Okay, one is biopharmaceuticals. So biopharmaceuticals is basically when you use biotechnology to produce medicines, pharmaceuticals. Okay. Now, there is one thing. There are a few key terms that are given in your notes with respect to biopharmaceuticals. So go through that. I'm not discussing this in detail in this lecture. Why? Because very little information is there and that can be covered through notes. But one particular development happened in 2025, which was a proud moment for India. In 2025, using biopharmaceutical expertise, India developed a new antibiotic called nephithromycin. You all know that today bacteria are becoming resistant to drugs. So there are certain forms of bacteria that are called, well, antibiotic resistant or AMR, antimicrobial resistant bacteria. This nephithromycin works on them, attacks them. So bacteria which are resistant to all forms of other drugs, they won't be resistant to nephithromycin and you'll be cured. Okay, so this is given, plus some examples of use of AI and 3D printing. Okay, this can be only covered through notes. It contains few basic trivia that you have to just remember. So go through the notes. All right, and that would be all. Today was a long lecture, but it was totally worth it because these are those topics which have a high chance of featuring in your exams. Therefore, memorize the notes well. Okay, and that would be all from today's, as far as today's lecture is concerned. So I shall be taking your leave with the promise of seeing you again with the next topic in this lecture series called Prelims Powerplay. Thank you, everyone.